Emma. Okay, so I think we are just about on time now. I can see there's some more people starting to join. So for those of you who have joined already, welcome to today's Microsoft Reactor session, which is on genomics on Azure. So just to give you a quick overview of the Reactor and what we do. So Microsoft Reactors are local community hubs where developers and startups meet, learn and connect. So we do this by providing training, such as sessions like today's, learning paths and presentations, plus meetups and other community events, all really related to emerging technologies. So there are physical reactor spaces all over the world, but we are now shifting to more of an online focus, as you can imagine. So for today's session, you are welcome to ask any questions throughout. There is a Q&A window for all attendees to drop your questions into, and we will make sure they're all answered by the end of the session. We're also recording today's session too, which will be put up on the Microsoft Reactor YouTube channel within about 48 hours of the session finishing up today. We're joined by three wonderful presenters, Mutlu, Roberto and Erdal, who will be delivering today's session. So without further ado, Mutlu, I'll let you take it ahead. Mutlu, are you able to hear us okay? I think you might be on mute.
Yes, sorry.
There we go.
Can you hear me now? Hi, thank you, Emma. I'm Mutlu Dorel. I'm a senior cloud solution architect and the Azure UK Genomics lead. As a CSA, I look after data and AI, DevOps and serverless technologies on Azure. My background actually is bioinformatics PhD. My PhD was about applying machine learning to solve computational biology problems. Before joining Microsoft more than 4 years ago now, time flies, I worked in bioinformatics and speech recognition companies. I invited two of my colleagues from the Genomics team to the session. Thank you for joining us today from Seattle, Roberto and Erdal. And good morning to you, as it's only 10 a.m. I think where Roberto is, and 8 a.m. where Erdal is. Hope you already had your coffees, guys. Roberto joined Microsoft late last year. He has a master's degree in biomedical science from Albert Einstein College of Medicine. And prior to joining Microsoft, he was managing bioinformatics support for customers utilizing long-read sequencing at PacBio. Dr. Erdal Coşkun is a data scientist at Microsoft Genomics. He has been working at Microsoft for about 4.5 years. He has a bachelor's degree in statistics and a PhD on biostatistics. His main area of expertise is applied machine learning and deep learning on high-dimensional genomics data. So today in this session, I'm going to give a brief introduction to genomics, as I understand not all of our audience are familiar with the topic, but nevertheless they are interested in deploying, scaling out genomics workflows on the cloud, on the Microsoft Cloud, Azure. I'll touch upon our partner ecosystem, briefly demonstrate a couple of solutions, and then we'll do demos. Finally, we'll finish with a Q&A for those of you who have questions. I'd like to start on why genomics before talking about genomics. So one of the key reasons why genomics is on the rise is that it enables precision medicine. According to a Nature article from which I took this image, the top 20 drugs used worldwide have 80% non-responders. That means that we are wasting a third of our global healthcare spending, nearly $750 billion per year on drugs that do not work. This is not necessarily because these drugs are inferior, badly designed, but mostly because they are prescribed to the wrong individuals in wrong doses or in the wrong phase of the disease. And precision medicine is going to be key to address these problems. Precision medicine is a medical model that proposes the customization of healthcare, with medical decisions, practices, and products being tailored to the individual patient. I want to tell you a personal precision medicine story very briefly. My wife had multiple miscarriages, unfortunately. Medical doctors could not tell us about the causes. They simply had no clue. Luckily, partly, say, due to my curiosity as a bioinformatician, I already had my genotyping done through a personal genomics company at that time. And I encouraged my wife to do the same test, which I did many years ago. My wife, who was working as a finance assistant at that time, believe it or not, started to read biology papers, talk in terms of SNPs, mutations, went through a lot of the reported SNPs, these genetic variations in her genome, cross-checked those against online information she can find in blogs, articles, and in even databases. Finally, the cause, or one of the causes, was there in front of us. She had a double mutation, or a homozygous mutation in a gene called MTHFR, which prevented her from efficiently metabolizing folic acid. Folic acid is crucial in pregnancies for the development of the embryo. The solution was simple: to take folate instead of folic acid, which MTHFR can normally convert folic acid into. We found other genetic causes, and I don't want to get into the details now. It's a long story, but we worked on a drug repurposing strategy to create our own combo drug treatment after sharing the results and our findings with multiple medical doctors and even in different countries. So it was out of effort, it was out of pain, it was out of effort, suffering, but in the end it worked. Not only once, but twice, as we now have a healthy son and a healthy daughter. And this wouldn't have happened 15 years ago. We are living in the future already. Precision medicine is not really a future concept anymore. It's already working, it's happening. And genomics is poised to transform healthcare by delivering clinical insights at population scale. So our efforts were personal, but the question is, do healthcare systems scale out such efforts? They should definitely, but currently, unfortunately, they can't, because genomics has been expensive and there are other various reasons. Now we are getting to a threshold where this is going to be possible. Healthcare providers will be able to leverage patient genotypic and clinical data together to tailor treatment to each patient, thereby improving and accelerating patient outcomes and reducing the risk of adverse reactions. So now, for those of you who are not familiar with the biology part, I want to give a brief introduction on that. DNA, deoxyribonucleic acid, is the chemical compound that contains the instructions needed to develop and direct the activities of nearly all living organisms. DNA molecules are made of two twisting paired strands, often referred to as the double helix. Each strand is made up of 4 chemical units called nucleotide bases, which comprise the genetic alphabet. The bases are adenine A, thymine T, guanine G, and cytosine C. Bases on opposite strands pair specifically, meaning an A always pairs with a T, a C always pairs with a G. The order of the A's, T's, C's, and G's determines the meaning or the information encoded in that part of the DNA molecule, just like the letters of a word in a sentence. And DNA sequencing simply means determining the exact order of the bases in a strand of DNA. In the most common sequencing technology used today, which is called sequencing by synthesis, we use an enzyme called DNA polymerase. It's used to generate a new strand based on the strand of interest. In the sequencing reaction, the enzyme incorporates into the new DNA strand individual nucleotides that have been chemically tagged with a fluorescent label. And as this happens, the nucleotide is excited by a light source, and a fluorescent signal is emitted and detected. The signal is different depending on which of the 4 nucleotides was incorporated. This is key. This method can generate reads of 125 nucleotides in a row and billions of reads at a time. So we check these signals, and we then know we can estimate whether the letter is an A or a T or a C and so on. And researchers can use DNA sequencing to search for genetic variations and or mutations that may play a role in the development or progression of a disease. The disease-causing change may be as small as the substitution, deletion, or addition of a single base pair, or as large as a deletion of thousands of bases. So, not all of the DNA parts are used to code proteins. In fact, only about 1.5% of our genome correspond to coding regions, and not all of the genes correspond to coding regions either. So basically, a gene is made up of exons and introns, and what happens is that introns are removed, and exons are glued together. Then this code is called messenger mRNA, and we send this message out of the nucleus. The original DNA stays within the nucleus. We send the mRNA signal into the cytoplasm, into the wild, where there may be viruses, bacteria, and so on. It goes to an organelle called ribosome, where protein synthesis occurs. So in a nutshell, DNA is first transcribed into an RNA. The only difference is between the DNA and RNA alphabet is the RNA alphabet has U instead of T. And then every 3 letters of RNA are read one by one, and they correspond to a single amino acid. GUG, for instance, corresponds to a V amino acid. And the amino acid chain, when complete, folds into a 3D structure and then we call it a protein, and the proteins are the basic units that do all the biological functions within our bodies. So this is the basic biology we need. So I already mentioned that precision medicine is a key driver for genomics, and cost is the second one. I think cost has been a key barrier to the widespread application of genomics in healthcare. When I was doing my PhD at the Sanger Institute in Cambridge, towards the end of the PhD, the price tag for sequencing a single human genome was $2 million just for one human genome. Today, it has fallen to around 1,000. And even more striking, at the beginning of the PhD in 2004, it was costing roughly 15 to 20 million just for one human genome. Last summer, actually, I managed to find a company in Europe, and I was able to do my whole genome for around 400, 500 euros, if I remember correctly. So the reduction in price is significant, and as a direct result of this, we see a rapid expansion of sequences becoming available in databases. In 2025, we expect around 100 million of genomes, human genomes only, to be available in sequence databases. This is excluding viral, bacterial, animal, plant genomes. So this is really an incredible growth, and that brings two challenges: storage and compute. We really need a new level of compute power to accommodate all of these sequences, all of this data. Sequencing a whole genome is going to generate around 100 gigabytes of data. Typically, this may increase. If you are doing a cancer genome, you sequence the healthy tissue as well as the disease tissue. You need to do it twice. Sometimes you need to do it over a longer time to watch the progress of the disease. And in addition to doing whole genomes, there may be other approaches that are currently popular due to the cost implications. So I mentioned about exons. The entire collection of exons in our body, in our genomes, is called exome, just a reference to the word genome. And that will only cost $200, and it will occupy 5 gigabytes of space to store. You may also do genotyping instead, only concentrate on interesting points in the genome where there are differences, between the reference genome and the input genome. Or you can do a gene panel in case you are interested only in a single disease, and that will mean that you are only sequencing a few genes. So in that case, the cost and the storage requirements will be trivial. But more increasingly, organizations, companies are doing whole genome analysis because they give you more information. So this statistics is at least 2 years old, but it gives us a good understanding of the storage problem, the scale of the problem in genomics. A single Illumina X10 machine is capable of generating 3 petabytes of data. 1 petabyte is 1024 terabytes every 18 months. And this was equivalent to the entire Netflix archive 2 years ago. I don't know the number today, but 3 petabytes in 18 months generated by a single machine. So it's really a huge requirement. So let's now switch the focus to what Microsoft is doing in this space. Let's talk about the genomics analysis workflow for a minute. So we start with the primary analysis. Primary analysis is about reading the sequence. I mentioned the Illumina Extend machine, for instance. There are other solutions, PacBio, Oxford Nanopore, etc. The first phase is base calling. I mentioned about signals. We need to convert these signals generated by the machine into a stream of A's, T's, G's, and C's. This is called primary analysis. And we store the letters in a digital file, digital format called FASTQ. Once we have that, we can align the sequence against a reference genome, and we record that, we save that alignment as a BAMP file. While doing so, we can produce another text file this time, which is called variant calling format, VCF, and that stores all the differences between the two genomes. And that's a text file. Every single line contains a variation, and VCFs are hard to read, especially when you have thousands of them. They are not ideal to do analytics on top manually. And tertiary analysis is anything you do downstream of secondary analysis. It can be about clinical reporting. It can be about doing GWAS, genome-wide association studies, finding links between phenotypes and genotypes. So Microsoft created a solution that is scalable, secure, and compliant. It's a first-party native solution on our cloud, Azure. It's accurate, easy to use. I'll show you in a demo very quickly. And it's transparent in terms of pricing because you pay basically based on the number of letters contained in the DNA. So let me now switch to this demo. I have a config file called demo config that I need to prepare beforehand. This is an API URL, and you need to also provide an access key. If you are wondering where you get these, you go to the Azure portal, and then you can create a genomics service. So let me quickly type genomics, and then you say create. It will ask you a couple of questions, and you hit create. I already have genomics accounts created. They are listed here now. This one, for instance, will tell you that the API URL is this West US2. You can copy it and you can put it in the config file here. And then the key can be found under access keys. You just copy it from there, and you put it in the config file. You specify the reference genome. This is the version of the human genome that we are comparing our input sequences against. You then provide the storage account details, an input folder, if you like, container in Azure terminology. And then the key to access that, you need to provide an output folder, which I called output. You need to put the input files, and then you can put a description. Once you do that, we have a command line utility called msgen, which you can install with pip. So if you are using Python 3, you need to put pip to install msgen, and it will install. I already have it. It's very easy to use. You say msgen list, for instance, and it's going to list all of my workflows submitted previously. And I say use this demo config file. This is going to list all the workflows. I have one that was cancelled. I have one that was completed successfully, and it will give you some details. If you want to get a particular workflow, you need to provide the workflow ID like that. W -W. If it's an ongoing run, it will tell you how many bases were processed so far and how many left. So this is the Microsoft Genomics service in a nutshell. It's really very simple. It's just addressing the basic secondary analysis problem. We also have a rich partner ecosystem. Here is a collection of them. One of the key things to highlight here is the breadth of different partners and solutions. Companies offering secondary analysis to tertiary analysis, those specializing on microbiome analysis or clinical decision systems. Now I want to talk about a couple of examples of partner solutions very quickly. A couple of years ago, Adaptive Biotechnologies, a front runner in next generation sequencing of T and B cell receptors of the adaptive immune system, announced a partnership with Microsoft to map the genetics of the human immune system, or immunome, another word, in order to detect cancers and other diseases in their early stage when they can be more effectively treated. By combining Adaptive's powerful immune sequencing technologies with Microsoft's research and large-scale machine learning and cloud computing capabilities, both partners aim to create individual disease diagnostics and ultimately a universal diagnostic, all from a simple blood test. While and then there came COVID-19. So Adaptive and Microsoft thought that this approach can be adopted here, and pretty much all COVID-19 research efforts look at the immune response. Looking at the immune response are focusing on the B cell, which produces antibodies, or they also look at the virus itself. This approach is different in the sense that it's focusing on the T cell. T cells are adaptive immune systems' first responders to any virus circulating in the blood, to detect and quickly multiply to attack the virus, often before symptoms appear. So Adaptive and Microsoft are generating years of data in just 3 months to accurately map the T cell response to SARS-CoV-2 or COVID-19 from initial exposure through clearance of the disease, covering all stages, and the data is available for researchers. And the purpose of this project is to determine these specific pieces of the virus our T cells recognize and respond to. You can read more on the blog if you search for the Adaptive Biotechnologies and Microsoft press release for COVID-19 research. Another example I want to give is about an open source solution. We also support the open source community, open source genomics initiatives. An example to that is OpenCB, or Open Computational Biology. So let me take one step back. There is a huge problem with VCFs, as I mentioned earlier. Each line in a VCF represents one genomic variation. Unfortunately, having such information in text files is not an ideal practice when it comes to analyzing and comparing different people's genomes, conducting research, or doing a clinical assessment. Just take the Genomics England's 1100 Genomes project. It produced 100K of genomes, and that equates to 1 billion genomic variants and 100 trillion genotypes. It's a huge amount of data. and you can't just store such information in text files to properly query it. And OpenCGA, a software tool under OpenCB umbrella of software tools, is a fast distributed open source query store analytics engine for genomic variants that eliminates the need for having to deal with individual text files. It's fully integrated with Azure services such as Azure Kubernetes Service, HD Insight, Azure Data Lake, Active Directory for authentication, and so on. It can be deployed with an Azure ARM template. ARM stands for Azure Resource Manager. And it's being used by the Genomics England research environment already. Large pharma companies are already using it on Azure. This is how the architecture for OpenCB looks like. IVA is the interactive variant analysis browser. It's the UI of this system of software tools. And Cellbase is which is here, a scalable high-performance database that integrates annotation data from well-known data sources such as Ensemble, UniProt, and so on. So it's used for annotation of the variations, genomic variations. And this solution comes with REST APIs, SDKs in Python, R, and so on, command line. Let me do a quick demo if we have time. So I'm going to log in to a OpenCGA instance. This is the IVA, the UI for OpenCGA. Here we are seeing 300,000 different variants, and it lists all these variants one by one in a table, giving us extra information such as the population frequencies of these mutations or single nucleotide variations, with some pointers to public databases and some annotations, whether it's disease causing, whether it's deleterious, and so on. So you can filter here on the right hand side according to genomic location, consequence type. Let's say I want to only list the loss of function terms, and I go and click and search. So now we are from 300,000, we reduce the search space to 1,000 almost records, and then I can click on a specific variation here and do more advanced stuff here. I can look at the annotations in other databases. You can also search by disease, because at the beginning of my talk I mentioned MTHFR. Maybe I can put MTHFR, or it can be any disease such as melanoma. Let's see if I do search. So this family or this individual has two actually mutations around MTHFR. One of them is deleterious and damaging with a high probability. So here under the Beacon tab, Beacon is a GA4GH specification. GA4GH is the Global Alliance for Genomics and Health. Microsoft is part of this consortium. It specifies the future standards for bioinformatics. You can search the Beacon network to see if this chosen variation is found in other databases. For instance, the Wellcome Trust Sanger Institute has it. It returned true. That means I can go and talk to them in case they have other phenotypes, if I'm a researcher or doing some clinical studies. So This was OpenCGA, OpenCB, and OpenCGA and OpenCB are supported by one of our partners, Zeta Genomics, which is listed here. This is something new. Very soon, we are going to make a few genomic data sets publicly available on Azure for ease of use. In your genomics workflows, application scripts that you are running on Azure, you won't have to download all of these data sets, genomes, reference genomes, and so on from external sources, but they'll be available through APIs, and Erdal is going to demo it after Roberto does a Cromwell demo. Cromwell, I'll come to that, is a workflow engine. So Azure Open Datasets is actually an existing service, and there you can find datasets on weather, census, holidays, public safety, location-related data that you can readily use in your machine learning experiments. Open datasets are in the cloud on Microsoft Azure, are integrated into Azure Machine Learning Service. They can be accessed through APIs, and you can use them in other products such as Power BI if you are doing dashboarding, or Azure Data Factory for doing data orchestration. I'll leave it to Erdal to explain more. So now I want to briefly talk about an interesting Microsoft research project. Genomics at Microsoft is not only limited to the Microsoft Genomics service or our partner solutions, but we also do research thanks to Microsoft Research. They collaborated with University of Washington, and they have successfully demonstrated the first fully automated system to store and retrieve data in manufactured DNA. So this is what I call the opposite of storing DNA on the cloud. This is about storing information in the DNA. If you compare a flash memory with a bacterial DNA-based storage system, a USB disk is going to be good for about 10 years, at least, for data retention. Whereas if you store the same information in bacterial DNA, you'll be good for at least 100 years and maybe 500 years. And if you want to read more on this, there is a blog page, actually, if you search for Hello DNA storage, Hello Data DNA storage, you can see it. Now I want to, this is my actually last slide before I hand over to Roberto. So workflows are so crucial in bioinformatics, in genomics. You have one software tool in step one, and then you go from step one to step two. Step two may then fork into two steps where you may need to run two different software tools, and then you merge the outputs of that, and so on. And every single step may need to scale out to hundreds of different compute resources. And you need to also think about how you merge the outputs of the individual steps. The output of the first step becomes the input of the second step. You chain them in different ways. And as I said, there are many alternatives on that already. To name a few: SnakeMake, NextFlow, Cromwell. And one of my colleagues, Andreas Worm, actually created, wrote this. Andreas Worm created this blog, nice blog about how to run SnakeMake on Azure using Azure Kubernetes service. And Roberto is now going to tell us about the Cromwell solution, yet another solution, yet another standard. We like this solution, we like Cromwell because it adheres to the GA4GA, GA4GH specifications. Roberto, feel free to take over.
Okay, thank you very much for the introduction. And so I'll be just briefly demonstrating the Cromwell and Azure platform, genomics platform offering that we've been developing at Microsoft Genomics. So as was been previously mentioned, the Broad Institute, which for those of you who might not be familiar, is a leading biomedical institute in the world that has been on the forefront of genomics and personalized medicine for over a decade. And so recognizing and the problem of trying to do informatics at scale and genomic analysis at scale, they ultimately decided to build a workflow engine that satisfied their needs. And so that workflow engine that would enable them to be able to build, automate, and scale and bioinformatics workflows at scale is called Cromwell and Azure. Is called Cromwell. We've adapted that and implemented Azure-specific portions to that workflow engine to be able to enable users to be able to deploy their own Cromwell instances on Azure and therefore be able to do high throughput, massively scalable genomic analysis on Azure. So first and foremost, one of the key advantages to utilizing this platform is that to get started is extremely simple. So we can actually deploy this simply using a simple command line executable that is available for all platforms, Linux, Mac OS, Windows. And so we have just simply have to download an executable and be able to run that in our and be able to run that on our system. And in one simple command, we automate the deployment of all the necessary resources that Cromwell will use in order to be able to actually then be already pre-configured to run our genomic analysis. So how does Cromwell know what analysis we want to do? Well, we specify that using what we call a workflow definition file, or a WDL file for short. And WDL files are a JSON-like workflow description language that allow us to specify how tasks are constructed and built. So we can see here, for example, that we have in our in a WDL, we can actually specify other WDLs where we want to inherit individual methods similar to, for example, class abstraction in Java. And we can actually specify what our inputs are, individual calls to methods within the WDLs that we need. We can parallelize tasks that can be parallelized. And ultimately, we can actually specify all the outputs that we need. This particular WDL is a best practice implementation of a workflow that will allow us to determine what is the difference between the human genome reference sample, which is the human genome reference that has been built and developed primarily through the international collaborations between the NIH, Sanger Institute in the UK, and others, and an individual sample. And we call this germline mutation identification. This WDL implements this in best practices and is already set for us to be able to run. So I can simply take this take a copy of what's called a trigger file, which we also specify in this repository. And by utilizing GitHub to maintain our repositories, we can maintain version and access control, I said, to these. And so I can actually take my trigger file, which you can see is pretty simple. All I specify is where my workflow is, where my inputs are that I need to specify, any options I may need to specify, and any external dependencies that WDL may have. And I can pass those to Cromwell. Passing that's pretty simple. As you can see here, I have the Azure Storage Explorer. And so when I run the Cromwell deployment tool, I ultimately have one of the things that happens is we actually deploy a series of blob containers that are tied to a virtual machine that is actually running Cromwell. And if I want to actually run this workflow, I can simply copy that trigger file and upload it into the new directory or the new container for this particular running instance of Cromwell in Azure. The trigger service will automatically detect that I have a WDL running. And at this point now that the trigger service interprets that workflow definition file and the individual workflows that we're trying to run and start submitting individual jobs to Azure Batch. I can track the progress of that here in the in progress container in progress folder within the workflows container. And when my job is done, I can go to the succeeded folder and see that my individual workflow is done. From here, so you can see that I've actually run this a few times in the past few days. I can actually go and using this unique identification number, this Cromwell ID, that is generated automatically upon submitting the job, I can actually navigate to that folder. And all I can see and look in to see all the individual steps, as well as the final results, which is producing my list of variants. Variants being those things that are different from this individual sample. This is a publicly available data set, NA12878, and can be downloaded from a number of different sources, including the National Institutes of Health. And ultimately, I can then look at this VCF file directly, as has been previously mentioned, or I can pass that VCF file into further downstream tools or build a database such as implemented in OpenCV to be able to actually combine those results with other samples and be able to do population scale analyses utilizing the results that can be scaled as necessary by utilizing Cromwell in Azure. Furthermore, this platform is extremely flexible because I don't necessarily have to do one particular analysis, but rather, as long as I have the ability to build a WDL file as a developer, I can quickly specify the individual tools that I need, and parameters, Dockerize them, and make them available to Cromwell to be able to run and utilize these tools. So with that, I'll hand things over to my colleague, Erdal, who will then dive into what happens downstream and further the further analyses of these files and some of the end-to-end workflows and such that he's been developing using he's been working on.
Yeah, hi, thank you, Roberto. So as we discussed with Mutlu and Roberto, we have different efforts on genomics. And one of the important component of these kind of genomics analysis is notebooks, because at the end you need to share your research, or you need to share your data science models with the notebooks easily. So today I'm going to show you the preview of the upcoming genomics notebooks that we will share with our stakeholders. The first GitHub that we share is the genomics notebooks, and this notebook repo will have different notebook cases, including and almost end-to-end genomics analysis with UBAM to structured data table analysis. And as Mutlu mentioned, we will provide the public genomic data sets in Azure soon, and you will see the notebooks that you can use for downloading and processing your data. And I'm going to show quickly for these notebooks. And the another option will be we will share our notebooks that you can use for the R bioconductor analysis. So we will have different notebooks that you can use for machine learning or the bioinformatic analysis. In this repo, you will see how can you create your own Azure machine learning workspaces for using the notebooks, or how to run your notebook analysis or notebook codes in the workspaces. And the first example that I would like to show you is we have a Cromwell on Azure instance and the repo that Roberto mentioned, and I'm going to show you the first case, the first notebook case that you can use for germline alignment and variant calling analysis. In the first notebook that we will share, you will see 11 different steps for doing the germline alignment and variant calling analysis, plus using some open source GATK functions. All these notebook solutions can be used on different environments like HPCs, like the Spark, or in your VM. This means you are not dependent on the specific environments. But in this case, you will see that how can you deploy, download and deploy your Cromwell on Azure instance from your notebook. And once you deploy your Cromwell on Azure instance, you need to execute your pipeline with a trigger file. Then you will see how can you upload your own middle file, own input JSON files, the input parameters that you need, and a trigger file. The only thing that you need to use is the SAS token that you have from the Cromwell on Azure instance. Then you can use your notebooks. You don't need to leave any the other tools, and you can continue to create your instance, upload your specific configuration files, and trigger your pipeline in the Cromwell and Azure. And once it's ready, you can download your results from the same storage account in your Cromwell on Azure instance. And once you downloaded the file, the output file, let's say in our case, this is a very well-known public data set, it's the NA12878, and you can continue to do your analysis with GATK or any pipeline or any Python libraries or R libraries. So in this case, we will add some public functions like annotate your genotypes using variant filtration in GATK, select specific variants, transform your filtered genotypes in a specific calls or no calls. And if you would like to do some concordance analysis, definitely you can use the GATK functions in this notebook too. And if you would like to merge the multiple VCF or GVCF files, definitely you can merge it from your notebook. And once you did all these filtrations, all these analysis, definitely you can convert your VCF file to a specific structured data frame, it's a data table. And once you have your data table, definitely you need to visualize it in a different tools. And we created a specific template, the Power BI template, that you can download and import your structured data frame on your cloud Power BI account or your client on-prem client. So this is like almost an end-to-end genomics data analysis notebook, from creating your Cromwell and Azure to create your own Power BI visualization. For the public data sets, you will see the notebooks probably very early August, and you can easily import your public data sets, like say CleanMar, to your environment that you run your notebook, or download a specific file. Definitely you need to know where is your data in Azure, but we will share all these details in the public data sets repo soon. And it's same for the reference genomes, because many people need a reference genome for doing benchmarking results, or sometimes checking their concordance analysis for the different pipelines. So we will provide the reference genomes, and you can definitely use notebooks for calling this data and importing this data to your environment. Quickly, I would like to mention that you can use the machine learning, Azure machine learning, for using these notebooks, because we have a Jupyter lab and we have Jupyter notebooks in Azure or RStudio. So you can use all these notebooks in your secure platform, Azure, because at the end, your data will be in Azure and your notebook will stay in Azure, and you can definitely use the Jupyter Labs in Azure for using different notebook cases that we will share. And I would like to share that you can use Python or R kernels in the notebooks in Jupyter Labs in Azure, so you can easily combine your Python and R functions on the same notebook and share with your stakeholders. And my final probably comment is please follow our announcements on the public data sets and the Genomics notebook repo, and we will be happy to share all the new notebooks this summer. Thank you.
Okay, Mutley, did you have anything you wanted to share before wrapping up the session?
I just want to actually, before we close the session, I want to thank Erdal and Roberto for joining us at such an early time from the US. And I want to share my screen so that participants can see the link to this survey. Let me know if you can see it.
Yes, it's there now.
So there have been some questions which we were trying to address. There are quite a few. So was Microsoft's involvement for the research in genome sequencing was provisioning compute and storage solution? No, it was more than that. We contributed to the research, and also we did a prototype. We created a prototype machine, an end-to-end system, if you like, that can take an input word such as hello and then encode that into a DNA sequence and then synthesize it. So don't forget that Microsoft is not just about Azure, but we also have Microsoft Research, as I mentioned before. We have a synthetic biology department as well in Microsoft Research Cambridge. And there is a team working with the University of Washington team on this subject. Roberto already replied to the question on the SV pipeline. So yes, please get in touch with Roberto on that. Why Azure or AWS or GCP? This is a generic open-ended question. I mean, cloud is great. Doing genomics on the cloud, first of all, is the way to go because there is a scale issue. Each cloud will have its own unique features, strengths, and Azure is well established, especially in the enterprise, and we have more data centers than GCP and AWS combined, so availability of the data centers is key, and we have also more security certifications, compliance overall compared to the other clouds, but it's really a bigger discussion to have. It's not an easy question to answer. So thank you all. I'll try to go over the questions. We overrun. I think it's already 5:01. So thank you for joining all. I hope you found it useful. And I want to also tell you that I'll be doing another session next week on Monday, this time covering Azure Machine Learning, how we create ML models, how we deploy them, how we scale them out, and also how we do inference, the entire end-to-end life cycle of machine learning model creation. Thank you very much for your time. Have a great day, evening. Take care. Bye-bye.